to the week 3 our of our workshop. All right, we've got one more. That's great. So I am gonna start off here with just a little introduction to the course. Okay, so for those of you who are new to the course this week, I know we had some that were who hadn't done this before. I want to go through some of the setup of the files that we'll be using and GitHub to facilitate you being able to work alongside with me in terms of the data and the programs and everything. If you have been here some of the weeks before, you'll have to do this as well. But we're going to find my GitHub website. So you can just type Alex McLean GitHub. Okay, let me share my screen here. That would help. Thank you, Elena. Great. Okay, so Alex McLean, GitHub, you can Google search that, and then click on the link, and then click on INBRE BIOS Summer 26. Okay. Yeah, oh great. So somebody added the link to the chat. Very smart. And that will take you to this page here. Click on this down arrow for the green code part, and I think it's easiest if you just download this as a zip file. So you can click on that, and it will download all the files that are in this repository. This has been updated, excuse me, with all the week 3 material, and you can put this on your desktop, your downloads, your documents, whatever file that, however you prefer, wherever you prefer to put it. So if you're on a Mac, you just want to double click the zip file. If you're on a Windows machine, right click and say extract all. I'll give you a second to do that. After you do it, can open it up, and what you want to get. So if this is your first day doing this, if you haven't done anything, then you don't have to do anything else. You are ready to go. All the week 3 bioinformatics stuff will be down here in this week 3 file. We can go into day 11, for example, the slides. These are the slides that we're going to go over. This HTML file. This is something that we're going to use as well as a companion document to the slides, show us how to do some of this stuff in R. If you have joined us previously, I would copy this whole week 3 file, copy it, and go to wherever you have your INBRE BIOS Summer 2026 files and just paste it into there. So if you have done this before, you would have week 1 and week 2, but you wouldn't have week 3. So just paste the week 3 stuff in there. And also there's this day 15 capstone project document. You can copy that as well and put it into your files. So I'm going to give you just a few minutes, 30 seconds to a minute or so. I just want to make sure everyone has the files in the right spot, or everything makes everything a lot easier. Okay. Everyone doing okay with this? Hopefully. Okay. Okay. All right. Thank you, Chad. All right. Hopefully everyone's good. Okay. All right. Okay. We will start from there. I'll share my screen again. The first thing I'm going to go over is I want to talk about the syllabus first, um, and particularly what our goals are for um, week 3. So I'm going to scroll all the way down to the last page here. Okay, so these are the week 3 topics that we will discuss. Today we're going to talk about genomics data, the structure of it, the format of it, some public databases that we can access. In the afternoon, we'll get into a little bit about accessing some of the public genomics data. Tomorrow, day 12, we're going to go through a differential expression analysis in more detail, talk about all the models that will run, what are the statistical properties of those, and why we get the results that we do. A lot of this is just very high level. So the functions that we'll use kind of do all the nitty-gritty details, and we won't talk too much about those nitty-gritty details on day 12 tomorrow. We'll get to those sort of day 13 and day 14 because there is a lot behind there. So first, we're just going to take a look at the full workflow from beginning to end. Day 13, we'll start to talk about some of those details in terms of multiple testing, false discovery rate. We're also going to talk about clustering on day 13, because that's one of the big things that happens from there. Day 14, we'll talk about dimension reduction. We had penalized regression on here as a possible topic. I don't think we're going to get to that. We're going to focus more on the dimension reduction side of it and principal components analysis and how to do regression with principal components analysis. Friday is going to be the capstone project. So let's go back up to the part that talks about that. So well, we can just go to the document here. We don't have to go to this. So we have a document here in the GitHub folder, day 15 capstone projects, which goes over the project that we're going to have for this week. And really, here's an opportunity for you to apply the statistical methods from the course on a real data set that interests you. This could be something that you're applying the methods we talk about in week 3, but it doesn't have to. You could be talking about the methods from week 2 or week 1 even. But the goal is to just do a small analysis, reproducible analysis, have a quarto document in the end that you'll present the findings from your group. You can work with a group on this. And because of that, after we go through this, I'd like everyone to introduce themselves a little bit. That could help you guys out with finding appropriate group members. Can have at most 3 participants in your group. The data set should be appropriate for at least one of the statistical methods covered in the course, and we'll talk about those below, some of the requirements. Here's some sites that you can use to get data sets. There's a lot of data on the web that is publicly available. These are just some places you can look. The NHANES data set, NIH dbGaP, does require application. This one is, these are a little challenging. I probably wouldn't recommend doing those unless it's something you're really familiar with. UCI Machine Learning Repository. This has hundreds of data sets, all of them really readily available to analyze. Bioconductor, experimental data packages. We'll use a lot of those this week. Tidy Tuesday data sets, Framingham Heart Study, which we've used through the course. You could use that as well in your analysis. So some of the checkpoints for the project, just to help you kind of keep moving on it. So by Tuesday, this should say day 12. At the end of the day, what is due? Just, hey, what is your group? What's the data set that you're going to be working on? So just, okay, let's have a checkpoint to where, okay, decided on the group, decided on the data set. By Wednesday, email me who the group members are in your group and a one-sentence description of the data set and the planned analysis that you have. So what kind of planned analysis are you going to be looking at in your project? And Friday, at the start of the afternoon session, you'll have hopefully have your rendered HTML report submitted, and you'll have just a short presentation describing what type of analysis you did, what the goals of it are, and what the findings were. So and then some more stuff about what to turn in on Wednesday. So some of the requirements. Two main types of analysis you could look at. One is an exploratory analysis. Here you don't have to have an outcome of interest or really an analysis of interest. It's just you are exploring data. So, just summary statistics, data visualization can be as simple as that. So correlation analysis, principal components analysis, that we're not going to talk about principal components analysis till Thursday, but if that's something you're interested in, we could work with you to help you get that part of it. Hierarchical clustering. Again, this is something we're going to talk about on Wednesday. So that certainly could be one that you do. Category 2, this is where you do have an outcome of interest. So you're either interested in the association of the outcome with one particular variable. You have one exposure of interest, you have an outcome of interest, or you're interested in prediction. And the type of, so that's what it means by inferential or prediction model. Inferential, this means you have an exposure of interest. For example, we are interested, does BMI relate with systolic blood pressure? Or predictive modeling, this is when, all right, we're interested in predicting systolic blood pressure from the data that we have. And this could be linear regression, logistic regression, survival analysis, and then the stuff we'll talk about this week, the differential expression analysis with DSeq2. Just quick structure of the report, quick introduction, 1 to 2 paragraphs describing the data, the biological public health question you addressed, why is it interesting, some description of the data set, just basic descriptives, clearly stating which methods you're interested in applying, applying your results or applying your models, figures, tables, then discussion. Hey, what are your results in plain language that anyone could understand? What did you find? What are the limitations? What would you do next if you had more time? What would you do differently? And similar to all the labs we've had in this course, we'll have a QMD file that will be what you submit as the final document. So to help you prepare for the final day, we'll have some short meetings. So that'll be myself or one of the teaching assistants. We'll send meeting links to you by Thursday morning. Those meetings could be on Thursday afternoon or Friday morning, depending on what works for you as a group. And so you'll work with us one-on-one to talk about your analysis, get some feedback on what you've been working on, troubleshoot any code issues you're having. This is going to work best when you come prepared with your data set loaded and kind of have some draft of your analysis underway. That's going to make the meeting most useful and kind of the most productive. Presentation you'll give at the start of the afternoon session on day 15. You don't have to have any formal slides or anything. You can just present the rendered HTML document, and just some things to cover in your 5 minutes or so that you present the findings. If it takes longer than 5 minutes, that's fine. Just what data set did you use? What was the question? What did you find? Walk through one or two key figures or tables. What was the hardest part? What would you do differently? Yeah, so I think, yes, submissions, email those to me. It's not going to be any, uh, grading or anything, just complete or incomplete. And this is what we're going to use for certificate purposes. So to get credit for the week, you need to have at least 75% of the material finished. The project will count as two. It will count as two of those, count as equivalent to two labs. So there is a total of six check marks that you can get this week. 1 through 4 will be labs, and 5 and 6 are going to be the capstone project. So some tips here. Start simple. Get a good simple question by Wednesday and kind of stick with that. I'll give you some feedback on how to implement your analysis pipeline here after you send me your document. So I always say this when I have students doing projects: keep everything simple. Just one simple question. That's so much better. One good, simple question is so much better. Even one okay, simple question is so much better than three, mediocre questions kind of done haphazardly. So depth beats breadth every single second, every single time. Write your interpretation and why you do the steps that you do as you go along. Don't leave that to the last hour. So that's going to be helpful for you at the end. Use the course lab files we've done as templates. So the structure and code are already there. So the solutions to the labs that we've done are all available on the GitHub, and you just downloaded them right now. So you can take a look at those. If you're stuck on the data, just go ahead and use the Framingham. You already know it well. We've used it in a lot of the labs. So certainly that's one you can do. And if you're more interested in the genetics stuff that we'll do this week, use some of those built-in data sets that we're going to go over today, tomorrow, and the next day.
Can only one person work on this, like as an individual, or do you want them?
Yeah, you can work on this as an individual if you like. Yeah. Okay. And if this is your first time, I think most of you, judging by the names, have been here already. Join our Slack team. You can ask questions on there, and if you're looking for a group, you could put something on there too. Okay. All right, so that's the main stuff, kind of preparation for this week. So I think with that, we can start with the material here for day 11. I'm going to share my screen, and I'm going to go to this, go to the day 11 folder. I'm going to open the slides and maximize those. And also, I'm going to have this ready. Some of this stuff. Get these. So um, if you have not done so already, there are some packages to to install here. So I recommend copying this and pasting it into your R and uncommenting these 4 lines right here. Um, these packages can take a while to load up, and check back on it as it progresses. So you could just go here, paste that in there, um, uncomment those, hit enter. I'm not going to do it here because I've already installed these. But this first part, this will install a package called BioC Manager, and it allows you to install packages from Bioconductor. That's BioC, stands for Bioconductor. And Bioconductor has a lot of packages that have to deal with the analysis of genetic data. I realize this is probably another one that you may have to install, this summarize experiments experiment package that you can install this one just using um, uh, install dot packages quote and summarize the experiment. So that one's a little easier. Let's see, we have a yeah, okay, there you go. And so do that, and that can kind of run in the background. At some point, it may ask you, do you want to update packages? They'll be like, oh, there's like these 10 packages you have old versions of. Do you want new versions of? It doesn't really have much of an impact one way or another in my experience. The updates to packages are usually things that I do not notice. But so you can update those, you can not update those, but that's the only thing that would it will stop there. So you do have to hit something one way or another. So check back over to RStudio to make sure it hasn't just stopped at that point. Okay. So what are we doing this week? So the next 3 days, we're going to move from just modeling a handful of variables, this situation like that we have with the Framingham data, a handful of variables on few thousand participants, to analyzing tens of thousands of variables simultaneously. So the biology that generates this data is a lot of the stuff. I'm very interested in some of the folks that we have here. You guys probably know so much more about this than I do. Um, we're going to focus on the statistical tools to analyze it. And the goal here today is to understand the structure of genomics data well enough to work with it with R. I'm not a bioinformatician, and I don't pretend to be one on the internet or anything. So I am a statistician, a biostatistician. So a lot of the upstream stuff is completely over my head. Um, and we're not going to really focus on the full analysis pipeline here, the alignment pipelines or call variants. We're really going to start where we have the analysis ready data. And hey, okay, how do we use that data to produce some analysis that will make some sense? The statistical challenges with this data, number one, many variables, tens of thousands of variables all at the same time. Number two, very small sample sizes. Three, correlated measurements. 4, multiple testing. We got about a half day dedicated to that on Wednesday. Those we will all talk about in detail. Okay, so let's talk about this typical, say, study here, typical RNA sequencing study, about 20,000 variables, observations, maybe 6 to 50. So one thing I'd like to say about these type of studies is, one thing I like to think about, what type of information are we getting, and where does this sit on the hierarchy of evidence? So the as the hierarchy of evidence goes, at the top of the hierarchy are meta-analysis. So meta-analysis is when you are analyzing data from, say, 30 to 50 clinical trials. So you're not only clinical trials are below that, below a meta-analysis, but so you're taking clinical trials, which are sort of the best thing. You have one hypothesis of interest. You're randomizing treatment, and you're looking, hey, is there an effect of this treatment or whatever you're looking at? Um, so clinical trials are right below that. And at the top, we have meta-analysis, which are the analysis of many clinical trials. So that's sort of the top of the hierarchy of evidence. What we have down here is not at the top. Okay, these are not things that will prove associations without the shadow of a doubt. This is closer to the bottom because here we have 20,000 variables we're interested in, and we have a few number of observations. Is this, are these do they tell us nothing? No, these type of analysis, these types of studies can be very valuable for, things that we just don't really know yet. I mean, these are sort of the beginning analysis that we need to do in the scientific pipeline. We always have to start from this point. Okay, we have a ton of variables. We don't know which one it is. We're kind of throwing everything against the wall and seeing what sticks. And then we'll go to the next study where we'll look at some of these things more in depth. And then the next study we'll look at, and then finally, maybe way down the road, we get to the clinical trial side of things. So these are hypothesis generating studies almost. So we have a ton of variables. We're not sure which of these genes really have an effect in what we're looking at, and we're sort of we're interested in kind of getting some initial information on this. Hope that makes sense. So what we call this in statistics, the P, and we use two greater than signs. N just means P is much larger than N. Whenever P is larger than N, we cannot do all the standard methods that we use pretty much go out the window. So we can't do linear regression anymore. Linear regression, the estimates require an inverse that you cannot calculate when P is as large as the sample size. So a lot of the standard methods get thrown out of the window. Well, what type of methods we need? We need methods that test each gene separately, then correct for what's called multiple testing, which we'll talk about on day 13. Account for the count nature of the data, which we'll talk about today, and then handle specific variant structure of the counts that we'll look at. So this data is a little different than data that we've used in the first 2 weeks. So the statistical framework that we'll go over in days 12 and 13 is it's more tractable than it sounds. So we do get very good information from these studies, but we really have to understand the data structure first, which is what we'll talk about today. So here's a little bit of the genomics data pipeline. And I know some of you know this stuff much better than I do, so I'm almost embarrassed to be talking about this. I'm among the experts we have in the audience here. Um, and really, there is a lot that goes in to to this analysis before we get to the data that we are really gonna focus on. So the first one that we may really focus on here is the FASTQ file. So the FASTQ, this is raw sequencing output, contains DNA sequences, quality scores per read, very large, gigabytes per sample, not human readable in practice. This is kind of one of the early stages of the genomics data pipeline. After that, there's an alignment that happens, and then we get the BAM files. This again, this is sort of more upstream than we're going to deal with, not anything that we're going to touch. After that, the VCF, which stands for variance call format. This lists genetic variants versus the reference genome. More relevant for GWAS, which we're not gonna go over, but that's another file format that you could see. Really here, as I said, we're going to work at the top right where we're going to get this count matrix, which we'll talk about next. These other ones, they require specialized bioinformatics software to be done right, which is beyond my pay grade here. So let's talk about this count matrix. When we have the count matrix, which we'll see, which we'll go to R to look at this in a couple after a couple slides here. The rows are going to be the genes, so 20,000 to 60,000 in a human data set. The columns are going to be samples. So you see in this example right here, I think there's only two different samples per treatment, so control versus the EXP. So not going to be a ton of columns here, but those are the different samples that we have. And the values we get are non-negative integers, and they correspond to how many sequencing reads map to that particular gene in a particular sample. So those counts, those non-negative integers, they make it so we want to use different types of statistical models than we've been talking about so far. The main reason for that, there's two main reasons. One, we have a small sample size. Okay, we have a small n. So this n is too small to assume the central limit theorem applies. So we have a small sample size. Second, the distribution is very right skewed. So a small number of highly expressed genes have very large counts. Most genes are going to have much lower counts. Many genes will have zero counts. So this makes it so that the t-test, type of idea that we've been using so far is not going to be appropriate for these type of situations. Variance is not going to be constant. The highly expressed genes tend to have much higher variance, something called over-dispersion. So again, standard linear models, t-tests to the raw counts are not anything we want to mess with. Even after a log transformation, that's not anything we really want to mess with. So here's a look at the distribution of the counts, and this distribution is actually on the log base 2 scale. So we see tons of observations down here. These are the zero counts. And then we have some very, very large counts when you think these are all on the log base 2 scale. So when we talk about count data, non-negative integers, there are two main distributions that come up. One is the Poisson distribution. one is the negative binomial distribution. They only differ by one parameter. So the negative binomial distribution has one additional parameter to it that the Poisson doesn't have. So the Poisson distribution has this assumption that the variance is equal to the mean. So as the mean grows, the variance grows. That's good. That is a good assumption most of the time. The problem is in this situation here, the variance is actually larger than the mean in a lot of situations. That's where we switch to the negative binomial model. So the negative binomial is similar to the Poisson, but it has this alpha parameter. When this alpha parameter is greater than 0, the variance is greater than the mean. So the variance, this mu is representative of the mean. The variance is equal to the mean plus alpha times the mean squared. So alpha is going to be that extra parameter that the negative binomial has that the Poisson model does not have. So the negative binomial is called an over dispersion model. Over dispersion just means extra variance, extra variance versus the Poisson. How this dispersion parameter, this alpha, is estimated is something that we're going to cover on day 12. I think it's super interesting from a statistical standpoint how this parameter gets solved. Because remember, we have 20,000, 60,000 tests, and we have this dispersion parameter for each one. It gets challenging to estimate that, and we can have a lot of variability. We can have wacky things happen. Okay, let's go over to our HTML file. If you want to open the .qmd, I encourage you to do that, and get this get everything squared away here, and run that first chunk of code. Get our data squared away. Okay, a little introduction. For all the morning sessions where we're going to talk about this analysis, we're going to use the airway data set. This is an RNA sequencing experiment measuring gene expression in human airway smooth muscle cells treated with dexamethasone compared with untreated controls. So this is, again, human airway smooth muscle cells either treated with dexamethasone or untreated controls. I know zero about the science of this data set. All I know is we have a treatment, we have a control, we want to compare those two. That's kind of the main statistical question that we have. This data set is not huge. There are bigger ones, but this one's small enough so that we can explore it more interactively, and it's not going to bog down our machines too much, and it's clean enough to illustrate some of the core ideas that we have. So we'll start off here by looking at the data set. And again, this is very different from the Framingham, where we had over 4,000 participants and we had fewer than 20 predictors. So this one really flips. So I use the data airway, dim airway, and we have 63,677 rows. Again, the rows are the genes, and we only have 8 samples. 8 columns of data. In a typical study, yeah, we could have 20,000, 6 to 15. Yeah, either way, we have much more variables than we have participants. So this creates a lot of problems. We can't do regression, just standard regression analysis. It's kind of completely thrown off. We have to test each gene separately, one at a time, which creates some some challenges to it. So we have to test a lot of hypotheses at the same time, which means we're likely to make a lot of mistakes unless we're careful. So we'll talk about multiple testing on day 13, which, okay, how are we careful? As we talked about, these data are counts. They're not continuous measurements. So t-test, linear models, we can't use those. We have to use other methods. We're not going to talk about the bioinformatics pipeline that happens before that. It's all very important, and I encourage you to lean into that if that's where your interest lies. Here, we're saying, all right, we kind of are starting from the count matrix. This is what we are going to analyze at the end. There's a lot of important things that happen with the FASTQ and the BAM and the count matrix that we get. So there's a long analysis pipeline that happens before we get the data that we get, but that's really beyond my expertise to talk about that. I can kind of talk about it from the point where we have the count matrix from there. Okay, well, let's look at the count matrix from this airway data set. So if I just print off airway, you can see that the class of it that we get, it's called a range summarized experiment. We'll talk about the summarized experiment class as down below. What do we get? We get counts, row names. There's 63,000 plus row names. So the row names are going to correspond to the these are an ensemble gene ID, these row names. We'll look at linking those into other information as we go on. The row names in the data, so we have gene ID, gene name, some others that we'll go over. The column names, these are the participants in the study, and these column names correspond to experiments that have different properties to them, for example, treated and untreated, so on and so forth. So let's take a look at the counts. We can see some of those right here. We have the gene ID, and then here is the sample that we're talking about. The statistical properties of the count data. Let's take a look at some histograms of the count data. So here we have the raw counts on the left, tons of zeros, right? And that may be collapsing even more than zero, depending on how big these bins are. And we don't see anything out here, and maybe like where's the data? It's there. It's just the bars are so thin that we can't even see them. Now this would be 5,000 right here. So if you just have one or two, yeah, you're not going to be able to see the height of the bar. On the right, we're looking at the same data on the log base 2 scale, and we look at count plus 1. We do count plus 1 because we can't do the log of base 2 of 0. So then we can see how many zeros, how many ones, and then going all the way out here. So those that are in this meaty region here, the actual values are 2 to the 10th, and then they only get larger from there. So very, very right skewed data, as we talked about in the slides here. That is pretty crazy how right skewed this is. Yeah, so the negative binomial distribution, which is generalizes the Poisson to allow for more variance, is one of the key mechanisms that we'll use to analyze this data. How many zeros do we have? How many zeros? How many that have less than 10? Quite a few. So 30,000 genes have zero counts. That means as we look across the whole row, as we sum across the whole row, so for each gene, we sum across the whole row, it's 0 for every sample, all 8 samples in the data. Here we have counts that are less than 10. So again, summing across the whole row, that there is less than 10 across those 8 subjects. So 30,000 with 0, so they don't have anything there. And then we have 41,000 that have fewer than 10. So most of the genes have fewer than 10, and almost half have 0. Um, just to give you a little indication of how many zeros, how many low counts that we have with this data. Any questions? Anything so far? Okay. I know you biology folks, hope I haven't can tell me all the stuff I've messed up later. Okay. This is one thing that confuses me, and I'd love to, if some of our biology folks would uh, could chime in here. I understand the issue from a statistical standpoint, but I'm unsure why it really pops up. So each sample is sequenced to a certain depth, so the total number of reads, and that's not equal across samples. So some samples can have many more reads than other samples. And if sample A has 20 million reads and sample B has 40 million reads, we'd expect B's counts to be roughly twice as large, even if the underlying biology is identical. So the raw counts that we were looking at, let's go back up to that count matrix. So these raw counts that we were looking at are all going to be relative to the number of sequencing reads that we have for each sample. And we can't directly compare those. So this is going to be a pre-processing challenge. So we are going to normalize for sequencing depth before we make any type of comparison. Um, this is something we're going to address on day 12 through a size factor estimation, um, which is a single number per sample that is going to count for the total sequencing depth. Here's an example of library sizes or the number of reads per sample. In this situation, we see some that have many more reads than others do, 10 times more. Um, in our experiment, um, we can look at this sequencing depth or library size, total number of reads. So we here we're looking at the column sums. So there's only going to be 8 of these. So there's 8 columns, and we're just taking the total number of counts in each column. That's going to be the library size or the number of reads that we have for each particular sample. And then we're just plotting them. And I'm looking at this here by treated and untreated. We don't want any relationship with treatment when we have this type of thing. And this is total reads in the millions. So the most is 30 million here. The smallest is around 15 million. So they vary across samples within the same experiment. And what I didn't understand, and if any of the biology folks could help us out, I don't understand why they vary from sample to sample. It seems non-intuitive to me, but again, I am just a statistician. I'm just here to correct for these issues. So, um, okay.
Yeah.
Yeah, someone gonna say something?
Um, I think because the input of the sequencing library is the different. I mean, even though you're trying to, before the sequencing and you construct the library, I mean, even though you try to, when you load down the sequencing machine, you try to look the same, but it's not going to be exactly the same.
Mm. So is it almost like some randomness that comes in?
I would think so.
Yeah, it's like you can't control
Yeah,
it.
you
as
can
to-
control exactly your load and how many copies of your DNA is in there. I mean, trying to
Interesting.
load the same, but I don't think it's possible to like get that calculation exactly. That's
Yeah,
from
yeah.
my understanding. Yeah, and also it's like from experiment to experiment. Like some people want to have deeper sequencing to get the rare express the gene. So they got more sequencing tabs. And then if my study doesn't need that, I can just do lower sequencing depths. And then when you compare these two, like higher one and lower one, and then you definitely have to adjust the library size.
Yeah. Okay. That's great. Thank you, May. That's wonderful. That explains a lot of it. Great. And here, what we have here, I think this is a good amount of how much this differs from sample to sample is for this airway data is kind of when you see this, you'd be pretty happy. Like, hey, this actually looks like it's pretty good. You're always going to see some differences there, and here what we see is kind of typical. Great. Okay. So it is the top of the hour at 11. I will give you a little break, a little brain break here. We'll come back and we'll talk about the different portions of this data in R and how to access them. So I will see you back here at 11:15. Let us continue on here. We are gonna go back to the notes. Okay. So talk a little bit about the organization of the genomics data in R and the summarized experiment object that we're getting. So there are three main objects that we'll be dealing with: the count matrix, which we just looked at, the sample metadata. So one row, this is going to have one row per sample. It's going to have the treatment group, patient ID, sex, age, whatever's in there. The gene metadata, so it's going to have one row per gene, gene name, chromosome, genomic coordinates, whatever it is. These three components, we have to keep these synchronized. They are separate. The summarized experiment object that we're going to be dealing with is designed specifically to keep these three things linked, which I think is really, really a nice feature of them. So here in some example data, so we have the gene. So the count data has gene down the rows, samples on the columns. Call data is going to have information about each of those samples, and then we'll have row data, which has information about each of the genes that we are interested in. Let's take a look at this, how this looks in R. Let's go to just click on the example 2 part here. So we have these three components, and when we have this summarized experiment object, these three are always linked, which is great. So how do we assess these three components? The assay, the count matrix, genes by samples, integer counts. This we are going to access with the assay function. So when we say assay, airway, here we're looking at the first 4 columns and rows. It gives us the count matrix that we have. So we can see some of those elements here. Call data, okay? So when we say call data of airway, this gives us the sample metadata from our experiment. And then lastly, when we say row data of airway, this gives us the gene metadata. So this will be all the information on the 60,000 or so genes that we have in our experiment. So how we assess these in R is we use the assay function, whatever our experiment is called, call data and row data. Now this doesn't work for every single experiment that has data in R, but this is a common format that is used, particularly with the data sets that you get from Bioconductor and some of the other places to get them that we'll talk about in a little bit here. There's also a metadata function that we won't go over. This stores experiment level metadata, who ran it, what was the protocol, so on and so forth. And we'll dive into these more when we get into the afternoon lab. Okay, so the call data, what is this? This is the study design matrix. It has the grouping variable for differential expression, any covariates that we may want to adjust for, and then batch indicators. And alignment is very important here. If the alignment of our call data is wrong or misaligned, every analysis we do from that point is incorrect. This is something that's easy to have an error with, and it's also pretty catastrophic because we have to do everything over again. And we'll talk about how the summarized experiment structure tries to prevent this from happening. First thing we want to do with this type of data is inspect this call data. So let's look at that more carefully. So I do call data airway. I'm just turning this into a tibble here. So what is the information that it gives us? Sample name. This is the sample identifier. Okay, we have all 8 of those. Cell, which is the cell line. This particular experiment has 4 different cell lines that we can see here. Okay, and then lastly right here. So this is a good place to stop and inspect this a little bit. So what's nice here is that every cell line, we look at it with both treated and untreated. So kind of gives us something about the study design of this particular experiment. It's good to see that. Next, we have the DEX, which again is treated or treated with dexamethasone or untreated control. This is the primary variable of interest. Albute here is whether the sample was treated with albuterol. It's not the focus here. All these cells are untreated. And then run, average length, experiments, so some other metadata on our variables. But if we had anything else like sex, anything that we may want to adjust for in our modeling, that would all be contained in this call data object. Again, the alignment is very, very important. So we want to make sure that the column names, the call data is aligned with the count matrix. So here we can get the column names of from the count matrix. Okay, we can see those there. And then the row names of the call data, which don't appear in the tibble objects, but they appear, they appear up here when we look at just the call data objects. And okay, we have both of those. Are they identical? Yes. Okay, great. So this is a good sign that not only they have the same call names, every call name in the count matrix is in the sample metadata, but they're actually aligned the same way. So we don't want to do anything until we do this, till we ensure that our data is aligned, because if we have any issues here and we just keep going, that's obviously can end up being a disaster. So one of the key benefits of the summarized experiment is that subsetting the airway object here is going to subset all three components at the same time. So they're all going to stay synchronized. So what do I mean by that? Well, airway is where we started. Okay, so when we get the count data, we use airway. When we get the call data, we use airway. When we get the row data, we use airway. So that's where we started. Okay, everything is in airway. Now here, what we're doing is we're changing airway so that we're only looking at the treated samples. And we're creating a new object called airway underscore treat, which only has the treated samples in it. So this changes from having 8 samples to having 4. What's really cool, let's look at the call data of this subsetted study, and you see that the call data of this only has treatment. So this new object we've created, it still has all three components to it. It has the count matrix, the call data, and the row data. And when we subsetted airway, all these things are still going to align with one another. So the call data here is still going to align with the count matrix. So all three of these things are linked in this summarized experiment object. So when we subset, we don't just have to subset one of them, then subset the other, then subset the other, and make sure they're all aligned. Here, we just subset the airway object, and then we use this call data, row data assay, and those are still going to be synced. So that, I think that is just a really cool thing that this has. And this goes for if we were subsetting samples or we were subsetting genes. It's all going to stay aligned with one another. So let's talk about that row data, that gene metadata. So here we are looking at the row data, just looking at the gene ID, gene name, gene biotype, some of the basic information that comes with this. What types of genes do we have? So here I'm doing a table on the gene biotype. Not looking at all of them. I'm just looking at a few, the top 10 here, and we see that most of the genes that we have are protein coding, but there's some non-coding RNAs, pseudogenes, and some other categories. So you may want to filter to only look at protein coding genes. You may want to filter so you're looking at everything else. And we can do this using the same way. So let's just just so we can take a look at this and make sure this is running with us in R. So here I've already loaded, I've said data airway, looked at airway. That's all we need to start here. And we can go down to this gene metadata and we can run this. We can look at some of the other information that we have in the gene metadata: sequence start, sequence end, sequence name, sequence strand, all kind of information. Yeah, and we can take a look at this more thoroughly. So you can see all the different types of genes that are in here. I just showed the ones that are the most prominent. Great. So the data that we are using is from the airway package, but there are a lot of other places that we can get published genomic data that's been deposited into a public repository. 3 of the main repositories: GEO, this is the Gene Expression Omnibus, the NCBI's repository for gene expression data, microarray and RNA-seq data. This is the one that we will use and demonstrate. dbGaP is another one. This is controlled access repository for data with individual level genetic information. So you have to apply for this and get IRB approval before getting data from there. Finally, SRA, this is going to give stuff more upstream data, FASTQ and BAM. And this would require significant processing, which is beyond what we'll talk about. So the Geo is kind of the right starting point for us, kind of give us some processed expression matrices and metadata that they're going to be immediately usable in R, and they're going to have this nice format that we've been talking about. So each study in Geo has a unique number to get it. We have the series, which is a complete study, a sample, so a single sample from within a study, or the GPL, the platform, the sequencing or microarray platform. So to use one of the published data sets, what we do is we find the GSC number in the paper's data availability section. Then you can download it with GEOquery in R. Pretty simple procedure. It downloads the data set for you. I did not use this for the labs because I just felt like it was another level that I didn't want to add on to this stuff that's already relatively challenging, but encourage you to play around with that, and we'll try to take a look at this if we can in the afternoon. And finally, there's the website here that lets you browse data sets before downloading them. So with these public data sets, the data provenance is going to be the documented history of how the data were collected, processed, and stored. And before analyzing any of them, there's some basic questions you want to ask to get from the metadata. What was the original biological question? What organism, tissue, or cell type were they interested in? What treatment and control condition are? Those are vitally important things, along with pre-processing batch effects we'll talk about. So you want to look at this metadata first to answer some of these questions, and hopefully they'll help you have some idea of whether it's going to be useful for you. What are batch effects? So these are systematic and technical variation, which can be introduced by when or how the samples were processed. These are what we call measurement error, and just these are measurement errors that come from a common source. So if we have different sequencing runs, different lab technicians, different lots, different dates, the errors in the data that we see can be correlated with one another, depending on if we know that, all right, these few samples were ran once, and then 3 months later we ran these other samples. There can be some batch effects that pop up. We want to make sure that these batches are not confounded within treatment groups. That's kind of a disaster, because then we can never differentiate the batch effects from the actual biological signal that we're interested in. So we want to check, okay, is there a batch variable in the call data, and is it balanced across treatment groups? So these are things that you want to control for batch effects in the design stage of an analysis. There are some methods to correct for batch effects. You see kind of some situations here where before batch effect correction, we can see clear separation between the two batches that were analyzed. After batch effect correction, you can't see the difference, but to me, you don't want this to happen. There are some statistical methods to correct for batch effects, but those are kind of things that it's like break in case of emergency in terms of using those. So we'll take a look at that in our in the R when we get back to the R document. All right, so what are some other quality assessment tools that we want to look at before having analysis? We talked about the library sizes. So are the total read counts per sample roughly comparable? Okay, yes or no? In this particular data, we see some variation here, but not too bad. Two, how many zero count genes do we have? These are not going to give us any useful information, and we really have to filter these out. So the genes that have zero counts, these are just, we can't ever say anything about these. So if there are, say, in this experiment, we know there's a lot, 30,000 of them. So we originally tested 60,000 genes. How many did we actually test? It was not 60,000. So there's 30,000 of these. We have to filter these out before we are going to do any analysis. So looking at how many zero count genes is important because it gives us some idea of actually how many genes are we really going to look at for differences between treatment and control, because a lot of these are going to be filtered out. Sample to sample distances. So do the samples cluster as expected by the treatment group and the cell types? We'll take a look at this in our airway data set. Are there any outliers? Is there any single sample that's very different from any others? One thing we'll talk about when we get to Thursday, day 14, we'll talk about principal component analysis as a data or as a dimension reduction method. So what principal components analysis can be used for? We're just going to talk about it at a very high level here. All right, we have 60,000 genes. Let's boil all 60,000 genes into two variables. And it's just like a quick visual check to see, hey, are things looking like we think they should look? And we'll see a nice example of this with the airway data set. They've looked very much like it seems like they should be looking. But here, for example, this a little bit of a warning sign here. So there's not much separation between the treated and untreated in terms of the principal components. So the untreated are a little bit higher and to the right, I would say. And the treated are more down here, but not great separation here from these. Also, we want to look for outliers. Is there one data point way up here? That would be a sign we need to take a look at that. We'll take a look at this. Let's go to our HTML file. First, this is something we looked at, the library sizes, and it's good to look at these by treatment, and don't really see much pattern here between treatment and control. That's a good sign. This next is going to look at the count distribution across samples. So a lot of code here, I realize, but it's going to create a smooth line that looks at what is the distribution of the count variable for all 8 samples in our experiment. And the color is by the treatment control condition. So we can see all samples, okay, they have a lot of genes that are in this very, very low count, 0 to 1 counts, and then goes down from there. And then all 8 of them kind of have this peak, I don't know, around 9 or so. Remember, this is on the log base 2 count, and then they trickle off from there. So the curves should closely overlap. If you have curves that are shifted or shaped differently from others, that is one indication that we could have an issue. How much is considered a departure? That's a good question. I mean, certainly we can see when it's very different. Here we see some differences, but I don't think any of these are any indication to be worried about that. So that's another quality control measure we can look at. So look at the library sizes, look at the count distributions to see if they're relatively similar.
Hey, Dr. McLean. We
Yeah.
had a question in the chat about PCA.
Yeah, okay. Thank you.
Yes.
Yeah, appreciate that. See if I can. Does PCA reduce the testing burden? Angel, that's a great question. So, and it's one that we'll talk about more so on Wednesday and Thursday. Yes, it does. The short answer, yeah, it does reduce the testing burden because we can reduce the dimensions of the data. We also could be throwing away meaningful signal when we do that. So yes, quick answer to your question, it does reduce the testing burden, but it can have some issues to it. Here, we're going to use this for for quality control more than testing. So we'll take a look at what we get down below. But good question. Yeah, sure. All right, next one, filtering low count genes. Here we're only going to keep genes with 10 total counts across all samples. So what's the idea of this? This is something we'll talk about Wednesday too. So really, if there's less than 10 total counts, there's really no chance that we see a difference. I mean, the extreme case of this is when there's 0 counts, we can't see a difference between treatment and control. So when we do this filtering step, we only keep situations where we have a decent number of counts and that there's really any chance that we see a difference between treatment and control. And this helps us in the multiple testing burden. So this is kind of related to Angel's question. This is another thing that helps us with the multiple testing burden is filtering some of the low count genes. Now, the number of genes filtered, the number of genes removed here, 41,000 genes are removed. So you may ask yourself, well, why use 10 total counts? Why not use 20? Why not use 30? The more we filter, the fewer genes we actually look at. So here, we're only looking at 22,369 genes. The other ones we never even tested. They were, in the study to begin with, but they never made it to the analysis phase. They were all sort of left on the sidelines. The rest of those genes, we can't say anything about them. So if your favorite gene was in there, we didn't test it. So there's kind of some pros and cons to this. It's definitely a good move. I mean, certainly, all those with zeros we have to get rid of. But it does create a situation where we are changing the number of genes that we tested. Next thing, so this is the sample to sample distances. So it's looking at how similar each sample is to one another. If the experiment is working right, we should see something with the treatment group here. Those should be closer to each other than other samples. So we're going to employ 2 methods here that we're going to talk about more later in the week. The first one is hierarchical clustering. So here we have a nice heat map figure, and we can see that when we group these into 2 groups, when we group these samples into 2 groups, all the untreated go to one group, all the treated go to another group. How dark the blue is here is going to be related to how closely related these two samples are. So it's darkest on the diagonal because that's one sample with itself, and those are going to be perfectly related. So we can see some darkness up here in the untreated group. Also see some darkness among the treated group, maybe to a lesser extent. And importantly, not a lot of darkness here between the treated and the untreated. And that's kind of a little bit of what we'd like to see when we look at this clustered heat map of the sample data. PCA plot is the next one. This is when we have employed PCA, and we've reduced this humongous 20,000 dimensional count matrix to just two dimensions. And we can see we have the principal component 1, principal component 2. So what we're looking at is the score for principal component 1, the score for principal component 2, it's going to be on the y-axis, for all 8 samples of this data set. So if it's positive over here, that means it had a high score on principal component 1. If it's large and positive here, that means it had a high score for principal component 2. What do these principal components mean? I can't tell you. All right, it's not really any. We will look at principal components and try to decipher what they mean, but that's not really the major point of this. Principal components is a way to reduce the dimensions of the data, and hopefully what we see makes sense. I think here what we see makes a lot of sense, and I think it's actually pretty incredible what we see here. So the red points are treatment, the blue points are control. So clearly PCA1 does a great job of differentiating treatments from controls. All the treated subjects have positive PCA1. All the untreated subjects have a negative PCA1. Further, we have the cell lines that we are considering. And remember, there's 4 cell lines in this experiment. I won't, you can see the names right there, but each cell line has a different symbol to it. And PCA2 seems to be differentiating one cell line from another. The 2 that have the lowest score on PCA2, those are the same cell line. In the middle, we have 4 separate samples from 2 cell lines. For the untreated, they're very close in their PCA2 score. Maybe the plus is a little bit higher. For the treated, the plus a little bit higher than the circle. And then finally, for the highest score on PCA2, we have the same cell line. So this is I think a great a great quality control device, and looks very good in this particular situation. That, hey, what we're seeing is what we would expect to see. Treatment, control, they're getting separated out. The cell lines are getting separated out. It's actually pretty incredible how well it works. I don't know if this is representative of how these types of things usually work. Okay, so the batches, the batch effect, again, that's a systematic technical variation that can be introduced when samples were processed. Some of this be available in the column data with the the run variable here. And we can tell that each cell line contributes, first off, each cell line contributes 1 treated and untreated. This is something that we noticed earlier on, that they broke down in this way. That means the treatment is balanced across the different cell lines that we look at. So that's a great experimental design. And then we have the run variable here, which I think is an indication of has some information in there about when this was run or which of these were run together. And we don't have any situations that these are the same. And I'm not sure about what these particular numbers indicate. You can tell that the same cell lines, the numbers tend to be more similar. So that means maybe those were run back to back from with another. Yeah, but I'm not sure about that. I'll have to take a more dig into the data there. Okay, so we're gonna use a different data set this afternoon and take a look at some of the same things that we looked at in the morning. Some of the key takeaways here from the morning. The data that we have are what we call high dimensional data. So we have many more variables than we have sample size. Creates a lot of statistical challenges to it. The analysis ready object that we are going to focus on is the count matrix, genes as the rows, samples as the columns, integer values as the data. It's very right skewed, small sample size. So we're going to use a negative binomial model when we analyze this in this data. The summarized experiment, nice object. We have the counts, the sample metadata, the gene metadata. It's all going to be synchronized, and we have some nice qualities to it where it stays synchronized as we subset the experiment. Always want to look at the quality control metrics first before modeling, checking the library size, zero counts, clustering, principal components, those kind of things. We want to look at the quality of the data before we dig in on the analysis side, which is what we'll talk about more tomorrow. The batch effects, these are something we want to control in the design, and we don't want these to be confounded with treatment. No statistical method can fix that. There are some statistical batch effects methods out there to control for them, but those are kind of break in case of using in terms of what we'll be interested in here. So I'm excited about digging into the afternoon lab today. We'll take a lunch break now. We'll meet back here at 1. And if you have some time, think about your project. Hopefully you can find someone to work with if that's what you're looking for. And have a good lunch. I'll see you guys back here at 1. Okay, so let's start the afternoon part. One thing I wanted to say regarding the introductions that we did in the morning, we will not include those on the clip that we put on YouTube. We had talked about that before the lab. Okay, let's talk about the afternoon lab. So we're going to use a different data set here. I can. Okay. All right. So we are this afternoon, we're going to go over some of the same quality assessment workflows that we did in the morning. Just going to do it on a different data set. The data set we're using here is the Pasilla data set. I think that's how you pronounce that. This is an RNA sequencing experiment in fruit flies cells. Comparing samples where the Pasilla gene was knocked down by RNAi, which I guess is an RNA interference with messenger RNA, to untreated controls. The Pasilla gene encodes an RNA binding protein involved in splicing regulation. So Again, I have no idea why this scientifically is of interest, but this was the scientific interest of this experiment. And this is one of the ones we were going to look at. This has a package which, similar to before, is available with this BioC manager. This package is on Bioconductor. And all these packages, you can always search for them in Google. And you'll see a little Bioconductor website pull up right here. You can always get some details about the data. This citation in Genome Research in 2011, which has some more details about this data set. Okay. So here I'm just loading in all the packages that we need. The Bacilla package is a little different than the Airway package. It ships with the count matrix and sample metadata as separate files. There is not a row metadata for this one. There's only count matrix and the sample metadata. We're going to load those in, and then we're going to create a summarized experiment object, because that's what some of the functions that we'll use, they will expect our data to be in that format. So here, this code right here loads in. Let's go ahead and pull up R here. What I have on here is still the stuff from the morning. See if I can find my mouse. Okay. I have all that loaded in. Now here I'm going to load in some of the next things that we have. So this counts file, this will actually point to somewhere on your computer, like this will be in the R directory. So this will find where this data is on your computer, and then these next two lines, they read that data in. So you can see the dimensions of the data here: 14,600 or so genes, seven different samples that we have. The names do not match here. So if we look at this, the metadata, you see it has this FB at the end. So those row names on the metadata do not match the column names. So let's look at the column names do not have that FB part in there. So this code right here is changing that. It's using the sub function. So it's subbing FB for nothing. And once we run that, the column names of counts will match the row names of meta. So let's take a look at the metadata. We have 4 samples that are untreated, 3 samples that are treated. The type column tells us whether the library was single or pair end sequencing. I don't know the difference between those two, but it's a potential variable that we may want to keep in mind when we're doing our quality control. In the untreated, we have 2 single reads, 2 pair end. In the treated, we have 1 single read and 2 pair end. This code right here is going to create the summarized experiment. So we just use the summarized experiment function, tell it what the counts are, and tell it what the call data are. And again, there is no row data here, so we skip that part. This next part is looking at the library sizes among the 7 samples that we have. relatively equal, not too much different between treated and untreated, which is good. There may be some difference by type, it appears. I think the single reads have a little higher total reads than the pair end do. So I think treated 2, treated 3, untreated 3, and untreated 4 are the pair end ones. Looks like maybe those are a little bit lower. And so there's 3 exercises here. These are going to follow what we did in the morning. The first one, we're going to extract the count matrix, print the first 6 rows and columns. What do the rows represent? What do the columns represent? Very simply, how many genes are in the data set? How many samples? So just some simple information. Look at the sample data. Okay, how many we have in each condition? Is the experiment balanced? Look at the type variable. Just some of the things we've talked about so far. Exercise 2, the first part is going to plot a histogram of the raw row counts for non-zero values only, and a histogram of log 2 count plus 1. Place them side by side using the patchwork library, describe the shape of each. So the first exercise of 2A, you'll want to go to this section 3.2 of what we looked at in the morning. And here, what we did was we got the counts for the first sample in the data. We filter it so we only have non-zero counts. Create the log 2 base 2 counts. For the second one, for the first one, we do not. And then this patchwork library is used to have both of these on the same in the same plot. So it's just asking you to reproduce that portion. How many genes have 0 total counts across all samples? What fraction of genes does this represent? How many genes remain after we filter them for at least 10 total counts? Then again, filter the SE object using just that, just those that have at least 10, confirm the new dimensions. The next part, you're going to recreate the density plot we had of the counts per subject down here in section 5.2, the count distribution across samples. For this new data set, are those two similar? Do they closely overlap like we saw in the morning? And the last exercise is going to have to do with the some of the PCA stuff that we did in the morning. I have some code here. You're going to use the filtered study. The filtered study is what we've created up here, where we filter to look at only those genes that have at least 10 total counts, and produce a PCA plot. And what do we think? Is there difference by the treatment groups in the morning? Does type appear to drive any of the separation in the PCA? So in the morning we had cell line driving the separation. Here we'll take a look to see if type drives any of the separation.